안녕하세요. 필코리아 테크놀로지 조은진 부장입니다. NGS는 이제 거의 필수의 실험 기법으로 사용되고 있으며 그중 마이크로바이옴 연구는 인간의 건강, 환경, 식품, 제약 등 다양한 분야에서 중요성이 커짐에 따라 이 NGS를 바탕으로 한 기술과 분석 솔루션 역시 지속적으로 발전하고 있습니다. 이에 오늘은 노보젠사의 마이크로바이얼 서비스 파트의 프로덕트 매니저이신 밸러리님을 모시고 Comprehensive Understanding Microbial Sequencing Solution 이라는 주제로 발표해 주시겠습니다. Hello, Valerie. Please share your information with us.
Hello, thank you very much for your kind introduction. So in this case, let me begin. Please give me a second. Okay, sorry. Hello everyone. My name is Valerie Joytan, and as for the kind introduction, I am a product manager primarily in the genome and the microbiome side, and I represent Novogene AMIA. So allow me to quickly share a little bit on Novogene. So Novogene is actually a global leader in terms of multi-omics solutions. We actually have 10 laboratories globally around the world, and we actually have many different services, which includes genomics, transcriptomics, single cell, spatial transcriptomics, metagenomics, and proteomics. And because we actually had 10 laboratories globally, so you can see these are some of our 10 laboratories around the world, and we also have 19 subsidiaries around the world. So for Novogene AMIA, we have laboratories within Singapore and Japan, but we also have labs around in Germany, in United Kingdom, the US, and in China as well. Because I represent Novogene AMIA, I would like to share a little bit on the Novogene AMIA side. So Novogene AMIA actually covers different accounts, around 400, more than 400 accounts and 10,000 projects from across Asia Pacific, the Middle East, and Africa. And this also means that we cover territories around Central Asia, which includes Kazakhstan, India, and so forth. So this means that we actually have many different types of samples across the world because we cover many different types of environments and also cultures. So these are some of our partners across Asia Pacific, which includes partners in Korea, in the Australia, Singapore, Hong Kong, and Japan. And one thing to highlight about our services is that because we offer a wide range of services, we also have many different platforms to meet this kind of needs. So this includes our NGS platform like the NovaSeq X Plus, as well as a long-read sequencing, which includes PacBio Revio and the Nanopore Promethea Ion. But besides that, we also offer other services, which includes mass spectrometry. So we have the Orbitrap Explorers for metabolomics and the Orbitrap Astra for proteomics. And to highlight across a point about multi-omics solution is that this means that we actually offer a wide range of services. This includes genomics, transcriptomics, microbial genome, which mostly means metagenomic in this case, epigenomics, proteomics, and metabolomics. Because usually for multi-omics solutions, it means that we're combining different solutions for the same sample. And I'll share a bit more for the multi-omics side in a little bit. But for today's webinar, I would like to focus mainly on the microbial side. As you can see, our microbial solutions are actually within our wide range of services, and this includes both the metagenomic and de novo sequencing. For today's intents and purposes, I'll be sharing primarily on the metagenomic side. So I'll be sharing on the metagenomic solutions. So our metagenomic solutions is part of our microbial solutions. Our microbial solutions is divided into two main sections. First, for metagenomic, which means mixed samples. This includes samples like stool samples, soil samples, and more complex samples like food samples as well. And for single bacteria and fungus, this means that we're working with single isolates, with either with a reference genome or without a reference genome. But for today's webinar, I'll be focusing primarily on the metagenomic side because for many of the metagenomic side, we actually do see a lot of applications within the multiomics region in terms of research. So for metagenomic solutions, we actually is divided between metagenomic sequencing and amplicon sequencing. And I'll explain a bit more on the differences between these two. So metagenomic analysis is actually, or solution is actually looking into the entire microbial communities. Most commonly, we're using shotgun metagenomic sequencing to sequence all the data within your sample. Another common method to look into metagenomic solutions will be through amplicon sequencing, which actually use targeted primers like the 16S region, the ITS region, and the ATS region to metabarcode and profile your microbial community. This means that there are some differences between these two solutions. For shotgun metagenomic sequencing, because we're using the entire DNA within your sample, so you're sequencing your whole metagenome within your sample. This means that you can actually look into taxonomy and function profiling, and you can actually do multi-kingdom detection. So within your same sample, you can look into things such as bacteria, archaea, fungal, protists, and viruses at the same time. And because you're usually sequencing at a deeper sequencing depth with shotgun metagenomic sequencing, this means that you can also look into a higher resolution. So we can actually go up to species to string level resolution with shotgun metagenomic sequencing. On the other side, we have amplicon sequencing, which does a targeted marker gene sequencing. This means that it's only meant primarily for taxonomic profiling in this case. And because it's done at a shallower sequencing depth, so you're usually looking into the genus and species level resolution at most. And because you're amplifying specific regions of your 16S or ITS or 18S region, so this is actually very PCR dependent. So it only does target specific detection in this case. Given the differences between these two solutions, this means that there's differences in terms of their applications. For shotgun metagenomic sequencing, a lot of the applications are done looking into the functional microbiome. And another big portion is actually looking into antimicrobial resistance, which is actually a big topic in terms of global health research, environmental health research, as well as the agriculture, because they're looking into antimicrobial resistance gene bacteria runoff from the animal husbandry all the way into the environment. Another key point for shotgun metagenome sequencing application is that you can actually do metagenome assemble genome or MAG reconstruction in this case. This is actually one of the end goals for most shotgun metagenomic sequencing. And as previously mentioned, shotgun metagenomic sequencing can also do multi-kingdom microbial detection. And because you're sequencing the entire genome from your sample, and if you're working with samples that do contain host DNA, you can actually look into host microbiome interaction studies as well. Amplicon sequencing on the other side is actually doing marker gene analysis. So the primary applications in terms of amplicon sequencing is looking into microbial community profiling. And so you're primarily doing diversity analysis and comparative microbiome studies. So a big topic in amplicon sequencing will be looking into environmental microbiome analysis, doing a quick community profiling of your different environments before you dive in deeper. So these are the main differences in terms of the metagenomic applications between shotgun metagenomic sequencing and amplicon sequencing. So let me dive a bit more into the services themselves. So for shotgun metagenomic sequencing, because you're using the total DNA from your sample, you actually only need a less amount in terms of your input. So for Novogene's recommendation, we require around 100 nanogram. And another thing to highlight about shotgun metagenomic sequencing services is that the analysis that you can do with shotgun metagenomic sequencing actually highlights its application. So you can see here in terms of its assembly-based analysis that we are looking into things such as taxonomy annotation, functional annotation, as well as antimicrobial resistance gene annotation. This aligns with the common applications that we see with shotgun metagenomic sequencing. If you're not doing an assembly-based analysis, what you can do is actually doing a mapping-based analysis. But again, you can see in terms of their analysis, we're looking primarily into taxonomy annotation and functional annotation. The analysis differs in terms of their databases, as well as how they handle the data, whether they do perform assembly or if they perform mapping-based. This is actually some of the data visualizations that you can obtain from analysis with shotgun metagenomic sequencing. For one, you can actually see that we're looking into the species abundance. We're looking into things such as the KGG function annotation. But another thing to highlight is that we're looking into things such as relative abundance of resistance genes, as well as annotated metabolic pathway. These are some of the analysis content that is more unique to shotgun genomic sequencing in this situation. Then we have the amplicon genomic sequencing services. So as previously mentioned, this is actually doing marker gene analysis with looking into regions such as the 16S, which stands for the microbial, like bacteria and archaea, 18S, which covers the eukaryotic side, and ITS, which covers primarily on the fungal side, as well as other customized region like the 12S region. Because you're doing a targeted approach with amplicon sequencing, you typically need a bit more DNA input. So we're looking into around like 200 nanograms for amplicon sequencing in this case. And in terms of analysis, you can see that the primary goal of amplicon sequencing is to look into its microbial community. So the analysis is primarily looking into alpha diversity analysis as well as beta diversity analysis. So alpha diversity analysis is trying to understand the community, the microbial community in this case, or if you're using 16S, the bacteria community within your sample, like within each individual sample. And then for beta diversity analysis, we're trying to understand the differences in terms of bacteria community across your samples. So alpha diversity analysis is looking within each individual sample, and beta diversity is looking across your different samples. And you can actually do 16S full length sequencing in this situation. And in this case, we're actually sequencing the 16S complete region, which goes from the 16S V1 to the V9 region. And we're using the PacBio review in this case for this sequencing. For 16S full length, because you're actually going with a deeper sequencing depth, you can actually go up to species level of potentially some strain level for the 16S full length sequencing. But again, I would like to highlight that you actually do require a bit more DNA input in this situation. So the DNA input that we recommend is around 300 nanogram in terms of Novogene's sample recommendation. But similar to the amplicon sequencing for NGS side, you can see that the analysis is primarily still looking into community profiling. So we're still looking into things such as alpha diversity analysis as well as beta diversity analysis. So you can see that in terms of their analysis content, there's differences between shotgun genomic sequencing as well as amplicon sequencing, whether with NGS-based or with 16S full length. And so here are some of the potential data visualizations they can get with amplicon sequencing. So we're looking into things such as alpha diversity, rare fraction curve, beta diversity rare fraction curve. And in terms of function, we're looking primarily into things such as function prediction. So we're not going into function annotation or functional profiling. A thing to highlight because amplicon sequencing is often used in terms of environmental research. So we do offer additional analysis such as network analysis for a lot of the environmental research in this situation. And so I'd like to move on to the next section, which actually looks into some of the considerations for multiomics. So multiomics is actually integrating different layers of biological information. And one thing to highlight is that when we're doing a single omics, sometimes it is limiting the data layer to only one fragmented view of the biology. So if you're only doing genomics, you're primarily looking into DNA variants in this situation. But it's kind of hard to see how this the entire biological system is interacting. And so you actually need to do a bit of a cross omics in this situation to understand the interactions in terms of regulatory networks that is invisible to the single omics side. So some of the multi-omics variation that we do see goes from genomics, transcriptomics, proteomics, metabolomics, and epigenomics all the way down. And actually, metagenomics plays a part in this role. So an example of a multi-omics study actually starts from the very initial study design. The study design, you have to go on from in terms of how you select your samples and how you group your samples. Because for multiomics, you have to consider potentially using the same sample or the same sample type for different omics consideration. What I mean by this is if we're looking into things such as diabetes and obesity, and we're looking for a specific sample, say a stool sample, we will have to consider things into how it applies for different omics. The first will be genomics. Are we looking into things such as GWAS, WES, or targeted sequencing? Or we can also do genomics sequencing for, if we're saying the host DNA for human in this case, we may be doing human whole genome sequencing. And then transcriptomics, where we're looking into the gene expression analysis. Are we interested in understanding how the gene expression is affecting in terms of the host for the human cells in this case? And we're also considering metagenomics. This is where metagenomics can play a part because the gut microbiota or the microbiome in different areas can affect different therapies and also how different diseases and interactions in terms of your environment. And then we can move on from there in terms of proteomics, which we're looking into the expression and modifications, as well as metabolomics, where we're trying to detect some of the smaller molecules in terms of different diseases. And by integrating all of this data, then we're looking into multiomics integration in this situation. So this is an example of a multi-omics study design because you have to consider many different layers from the same sample all the way to the different potential omics that you're interested in doing and how to integrate all of the different data. So an example of this is that we actually do offer an integration analysis with metabolomics because we're often trying to understand the changes within the microbiome and the changes in the host metabolism. So this integration analysis can be done with either the 16S or in this case, amplicon sequencing data or the shotgun metagenomic sequencing data. And we can combine the results, the analysis results with the metabolomics analysis results to understand more of a dual omics correlation analysis. In this case, given the differences in terms of the 16S or amplicon sequencing versus the shotgun metagenomic sequencing services, I will recommend the shotgun metagenomic sequencing plus the untargeted metabolomics if you're interested in some of the microbiome changes with the host metabolism. So these are some of the analysis that you may be able to get through this integration analysis. And so I would like to just highlight some of the case studies from our customers. So one of this is a case study where the customer is trying to understand the individual's biological age, their skin lipidome, and their skin microbiome. And so what the customer did is they actually collected samples from the skin tapes from different age groups across different genders. And what they found is that the microbial concentration between different age groups actually was quite significant and it was actually affecting the different lipidome or sebaceous gland activity. And so this can only be done in terms of the results by combining both shotgun metagenomic sequencing plus metabolomics in this situation. But when we consider multi-omics, this is not only limited to, say, a combination of two omics. When we talk about multi-omics, we actually see that you can actually combine more than one omics or more than two omics in this case. So this is another example where it's a combination between three omics, where the customer did genomics, metabolomics, and metagenomics in this situation. So in short, they basically found that the different inoculation of different microbials can actually reshape the metabolic pathways, the microbial communities, and the quality at a system level. So this is actually looking into the tobacco leaves. And so you will only be able to see this type of results by combining the three omics because you have to understand how it's affecting both the plant, the host in this case, as well as the microbial community and its subsequent metabolic pathways. So this is just a quick two examples in terms of how multi-omics are being used. And then last but not least, in terms of integrated multi-omics, we actually do see a lot of combinations that look into different biological functions, metabolic pathways, and regulatory networks. And this combinations is not only limited to metabolomics and metagenomics. We often see other combinations such as transcriptomics plus proteomics, transcriptomics plus proteomics and metabolomics, transcriptomics plus metabolomics, metabolomics plus proteomics, and transcriptomics plus epigenomics. So we do see a lot of advancements towards the multi-omics space. And so this is something to consider if you're interested in doing multiomics in the future. And that's kind of it from my side. May I ask if there are any questions? Thank you very much.
Thank you, Valerie, for your presentation.
Thank you.
Q&A 시간을 가지도록 하겠습니다. 화면 하단에 Q&A 아이콘을 누르고 채팅창을 통해서 질문을 주시면 우리 밸러리가 확인하고 답변을 줄 수 있습니다. 혹은 여기 손들기라는 또 아이콘을 누르면 저희가 발언권을 드리도록 하겠습니다. 아마도 이 대부분 이제 궁금하신 사항을 직접 말씀하시기보다는 채팅으로 글을 쓰는데 시간이 조금 걸릴 것 같습니다. Valerie,
Yes.
actually, I heard the multiomics in microbiome. So, what does multiomics mean in microbiome? And what type of sample should be spared for it? Prepared, sorry.
That's a good question. Thank you for asking this question. So, microbiome is used in primarily two methods in multiomics. The first is actually trying to do the metagenomic side, which is trying to understand how the microbiome community is affecting the individual of interest. So we often see this in terms of health research at this moment, and so an example of this will be for different obesity and other related health. They will often do metagenomics on their patients or usually patients in this case to do a time track of their patients from in terms of if they do different like surgeries or if they do different therapies, and they do a time track of when they started before the therapy, when they started the therapy and after therapy. So it's actually a big cohort study. They'll look into stool samples. Most of the times they'll take saliva samples, stool samples to track the gut microbiome in this situation. And then they also take potential like biopsy in terms, or they'll take serum or plasma for metabolomics, as well as sometimes they'll do mRNA on the human samples to look into if there's affecting any host gene expression changes as a result of this microbiome and as a result of therapy. So this is one of the more common multiomics that we do see happening in research at this moment, but it's not limited to cases such as the gut microbiome, because we also see many research that are doing into things like the skin microbiome, the eye microbiome, as well as other considerations in terms of the environmental side. But the environmental side is primarily looking into things such as the interactions that's a bit more complex, such as interactions between antimicrobial resistance with agriculture and with different runoffs in this case. So this is more on the metagenomic side, but we have also seen multiomics applications on the microbial whole genome sequencing, where they actually notice the application of specific microbes or like say fungals in this case, and then try to apply it to different other hosts. So sometimes they'll take the microbes, do fermentation, or they'll do infection, and then see how that interacts with the host, and the host response in terms of their metabolites and their protein changes as well. So there's actually a lot of different applications, but it's a combination of different solutions at the same time to answer one main question. Hopefully that helps answer the question.
Thank you.
Thank you. May I ask if there are any additional questions? I would like to highlight something in terms of the sample questions, what kind of samples should be spared. Usually in these cases, we see most often with animal-based samples such as stool samples, saliva samples, swab samples. But a thing to consider if you're using the same sample for different omics is to how you first collect your samples because different omics such as metabolomics sometimes have different sample requirements compared to say metagenomics as well as mRNA sequencing or metatranscriptomics. Because what we have found is that say if you collect your stool samples or your saliva samples, some of the stabilizing solution can actually interact differently with RNA versus the metabolites versus the proteins. So even with blood samples, if you're say want to do blood samples for different solutions, how you collect the blood, how you store the blood, can all affect which omics you can do in the long run. So usually, if you're interested in multiomics, I'll recommend starting from the study design to look into what kind of questions you want to answer, whether you want to answer gene expression, the microbial community, the metabolites, and then plan how you want to how you want to collect and store your samples, because this will affect all the different potential solutions that you may be able to use. Mm. Oh, sorry.
또 궁금하신 사항들 있으실까요?
Oh, sorry. I saw from the Q&A there's a question about um the sample requirements for amplicon and shotgun genome sequencing and the library type. So yes, the library type is listed for to be a single type. So what this means is that say I can actually share it really quickly here. I'll use shotgun genomic in this case. If you're working with a sample, I'll give a quick example. If you're interested in like I'll take an animal, a cow sample, which has been infected potentially with different microorganisms, and you wanted to do shotgun genome sequencing. What you can consider this is that this 100 nanogram is meant for the metagenomic library. But if you want to use the same DNA to do something else for, say, plant and animal whole genome sequencing, you may need to meet both data requirements depending on what kind of analysis you want to do in this case. So this is correct in that this sample requirement is listed for a single type. But if you're interested in doing multiple different library types, we may need to uh, add on the DNA. Another example for this is if you're interested in doing shotgun genome sequencing plus amplicon sequencing at the same time. This means that you have to have sufficient DNA for both library types. So this means 200 nanograms plus the 100 nanogram, so 300 nanogram in total to be able to do the two different library for the two subsequent sequencing. Hopefully this have answered your question. Thank you. Thank you. May I ask if there are any additional questions? Thank you.
음, 질문이 없으시면은 또 나중에 또 갑자기 번뜩 질문이 생각나시면은 필코리아 테크놀로지 혹은 노보진에 문의 주시면은 또 성심성의껏 답변 드릴 수 있도록 하겠습니다. 필코리아 테크놀로지는 노보진사의 한국 대리점입니다. 웨비나 이후에도 궁금하신 사항 있으시면은 언제든지 문의 주시기 바라고요. 마지막으로 저희가 설문을 받고 있습니다. 설문을 주신 분들에게는 또 저희가 소정의 커피 쿠폰도 제공하고 있으므로 많은 참여 부탁드리고요. 이것으로 오늘 웨비나는 마치도록 하겠습니다. 참석해주신 여러분에게 다시 한번 감사드리고 Thank you, Valerie, again.
Thank you very much. Really appreciate it.